out of an intercourse. And so remember we said when you undergo the second meiotic division, it actually has to bind with the sperm. Otherwise, it's released in this pattern as a cell structure. So we have the egg with its granulosa cells, the zona pellucida, small space between that and the membrane. The first polar body is still going to be present. So a series of steps occurs. It takes multiple sperm. That's why the minimum is about 20 million per mil, because not all of those sperm actually reach the egg at the same time. They will swim through the uterine cavity and will swim to the isthmus of the oviduct and then form a pool of sperm there. And waves of about 100 or so thousand sperm at a time will then swim up the oviduct and be propelled by the muscular contractions. And so if they do not meet the oocyte, then it stays as the secondary. If they do meet the oocyte, some of them will undergo the release of their acrosomal enzymes and digest their way through these corona radiata cells and the other cells surrounding the plasma membrane. A few of them will actually reach the zona pellucida and bind to it. And that binding is typically required for what's known as the acrosomal reaction. So some of them have this ability to undergo the acrosomal reaction, started, remember, in the tail of the epididymis. It actually changes a destabilization of the phospholipids in the membrane of the acrosome and of the plasma membrane over the head of the sperm. So if those destabilization changes don't occur in the tail of the epididymis, then when the sperm reaches the milieu, if you will, the environment in the female reproductive tract, and progesterone is one of the key requirements for that environment. The sperm lab over in Davis discovered that back in the early 80s or late 70s. Then the rest of that acrosome reaction doesn't occur. That process that finishes that and the ability for the tail of the sperm to start beating faster, the sperm wiggles its head really fast and its tail beats faster, that whole process is known as capacitation. So to enable that further steps of maturation in order to do that requires that environment of the female chemicals and hormones. And those were what was learned had to be added to the petri dish for in vitro fertilization. So let's take a closer look at what happens in the acrosome reaction. So here is the nucleus with the nuclear membrane, and this is going to be in chromosomal material of our late spermatid that developed into spermatozoans. And it's going to be surrounded by the acrosomal cap. You guys remember what the acrosomal cap developed from? The Golgi. Again, so on your exam, you will have a table in which I will list structures. What is the structure that's homologous to this? So the male structure that is homologous to or equivalent to the labia majora would be the scrotum. Okay? What does one structure develop from? So the cremaster muscle has its origin as the internal abdominal oblique. All right? The acrosomal cap would have its origin as Golgi apparatus. So this is in a longitudinal section. It's really more than just a U. It's like a knit cap that you would pull down over the head. And this is identified as the outer acrosomal membrane and the inner. And then around all of that, we would have the plasma membrane. Forming the head of the sperm. I mean, they're all a plasma membrane, but this would be the plasma membrane of the cell. So initiating the acrosome reaction is a fusion of this plasma membrane with the outer acrosomal membrane. The membrane breaks down. These two membranes break down and refuse with each other so that small sacs are formed. So this closes off here and closes off here. That gap is just for my line. And then these fuse together to form sacs. That's the acrosomal reaction. The fusion of the plasma membrane with the outer acrosomal membrane. And now if these sacs are no longer held to the sperm, they get flushed away. And the content, enzymes, digestive hyaluronidase enzymes of the acrosomal sac, then are now released. And that's what's going to digest the pathway for the sperm through the zona pellucida. So the next thing I'm going to do is erase part of these away so you can see what's happening. What initiates that? The sperm being within the chemical soup of the female reproductive tract. So if sperm are just ejaculated and deposited on a towel or whatever, they're not going to undergo the acrosome reaction.
And that's called the
capacitation is the changes that allow this to be completed, as well as the changes that allow the tail to be beat. So a acrosome reacted sperm then is going to have, if I can get the colors properly drawn here, the nuclear membrane, the outer acrosomal membrane, the inner acrosomal membrane, and then the rest of the plasma membrane. This is what contacts the plasma membrane of the egg. Okay. What initiates that that acrosomal membrane is when the outer plasma membrane of the sperm contacts the zona pellucida, and a protein on the zona pellucida known as ZP3, zona pellucida 3 protein. Okay? That's what triggers this to occur in most of the sperm. Some of them will do it early, and those are the ones that are going to kind of digest through the granulosa cells, but the rest of it occurs when it makes this contact here with the zona pellucida. So I have a phase contrast video of that happening. It's not, they disabled the embedding URL, so I'm going to go to it directly here. So what you're seeing on the surface is the changing, the formation of those vesicles from the outer plasma membrane to the outer acrosomal membrane. And these are the vesicles that are being formed as they fuse together. I'll play that again now that you know what you're looking for.
Oh, it was going?
So there's two things that are allowed to happen once this outer plasma membrane and outer acrosomal membrane are removed. Obviously, we're releasing the acrosomal enzymes, and we're also exposing the inner acrosomal membrane, which is what's going to actually bind to the egg. Okay. Surprisingly, what appears to be species-specific is the recognition of the ZP3 protein by the plasma membrane of the sperm. Once we get to the inner acrosomal membrane, that's no longer species-specific. So I suppose you could have a hamster sperm binding to a human egg. A hamster sperm won't bind to a human zona pellucida. Okay. From what I've read and preparing for the other classes, it's kind of like, oh, well, it seems to me both of you would want both of them for full protection. That doesn't mean you go on and form a new hamster person. The cell divisions have to be completed after that as well.
Can I ask you a question?
Yes.
what you had drawn in red. the
That
tip
was
of-
just the plasma membrane of the sperm.
Okay.
I mean, they're all formed of plasma membrane, but the surface.
Out of
Plasma
the
membrane.
membrane.
So this is an image of the same process. I have it on our schedule anyway, to watch, well, we can't see it to this level of detail, but to watch the fertilization of sea urchin sperm. Unfortunately, in our schedule, that day falls on Tuesday, which makes it impossible to get the sperm here, to get the sea urchins here from the coast and get their eggs and sperm out. So I'll show you images and videos of it, but to actually see it happen, unless they came early, we won't be able to do. We could make your test, 10 questions worth 10 points each for the 100 points and do this on Thursday, but with holiday being on Friday, anyway, I got an email just before school started saying that we couldn't get the sea urchins earlier than and able to do the experiment on Tuesday. But some of the processes we're going to look at. The lab for that day, however, covers implantation, which is what we're going to be talking about today and on Tuesday. So the lab itself the lab homework can still be done. So here are the cells of the granulosa cells, the corona radiata cells. See how these actually interact with the zona pellucida? Those corona radiata cells are intimately associated with the zona pellucida. So here comes the sperm. Its cells are going to bind to the ZP3, undergo the acrosome reaction, and then here you can see that it's lost its plasma membrane and outer acrosomal membrane as it's digesting its way through. And it actually comes in sideways. It doesn't just dive right into the egg membrane. It comes in sideways, and that's where it interacts with the egg molecule, the integrin alpha-6 beta-1 molecule. It's an adhesion protein. When that does that, it triggers the second meiotic division. So the membrane that the sperm is binding to is the membrane of a secondary oocyte. The binding of that sperm triggers an electrical current that initiates several things: a block to polyspermy from preventing other sperm from passing into the egg, as well as initiates the completion of that second meiotic division, so that the sperm head enters into an ootid. And we have a second polar body, so I'll show you that in a moment. So at the time that this sperm is trying to get through, thousands, hundreds of thousands of other sperm are also trying to get through, all right? One sperm, as soon as it binds to that integrin molecule, initiates 2 blocks to polyspermy. One occurs very quickly, and the other one takes a longer period of time. There might be additional sperm that bind to their own integrins, all right? But the fast block to polyspermy prevents any other sperm from binding to integrin. So it changes the electrical current of the membrane and changes the integrin molecule so that the sperm that haven't already bound to it no longer can recognize it. We still have a bunch of sperm that already bound to their own integrin molecules. So the second block to polyspermy, the one that takes longer, causes the zona pellucida to harden, as well as to separate, move away from the egg, and that kind of pops off any sperm that may have already bound to the integrin. Yes? What pops off? Sperm. So let me diagram it. Those two components.
I have 2 fertilized.
So we may already have, when this sperm binds to its integrin molecule, the sperm is actually going to insert its DNA into the cell. We may have additional, so I'll limit that here. So here's one sperm. That's bound to its integrin, okay? It initiates a opening of sodium ion channels, which causes a very quick depolarization of the cell membrane. While this one was binding, additional sperm might have bound to other integrin molecules. This one inserts its head and its DNA. Its tail remains behind. Tail doesn't come in. but we still have these other sperm that are bound to integrity. We don't want them coming in. So this first quick inflow of sodium ions is our fast block to polyspermy. It results in this spike of positive ions within the cell. We saw that with neurons, and we got depolarization of cell neurons. And so that prevents, it changes the shape and prevents other sperm which may not have bound from binding. Okay? So that first step of the opening of the sodium ion channels is known as the fast block. Now, it is rare, but it is possible for two sperm DNA to enter into the egg. All right? And if it results in the separation of two cells, then you would have non-identical twins. Most of the time, non-identical twins, like my twins that are a boy and a girl, you wouldn't believe how many times people would say, are they identical? And I say, well, no, they're brother and sister. I know that, but are they identical? Like, no, you want me to open up their diaper? They're not identical. So those are actually the ones that if families have twins, you know, it runs in the family. It's the non-identical type that seem to have a frequency that can be passed on. The identical twins we'll talk about on Tuesday occur when one sperm fertilizes one egg and early separation of the dividing cells occurs. Most non-identical or fraternal twins occur when you have two eggs released and a sperm fertilizes each one. I remember when I was in high school, we had to read Newsweek for our English class, and there was an article in there about a woman in Germany who had intercourse with two men within a 24-hour period, had released two eggs. One, they were both from the United States Army. One was white, one was black. And so she had, and that was the proof that she had two different fathers. And all that had to happen at the same time. She had to, you know, release the egg and everything like that. So obviously those were non-identical. The identical twin has the two identical or triplets that are identical originate from the same egg and the sperm. They share the same DNA. So it has to be the same egg that is dividing. All right, so fast block to polyspermy results in sodium ions entering and causing depolarization of the egg membrane. It's now an ootid. And a resulting change in the structure of the integrin so that it is no longer recognized by this other sperm. Yes, Tab?
Is the two sperm fertilizing one egg with chimeras happen?
It can be, yeah. It's more common like that way than, or the only other way would be for the other cells to fuse together, and that's not as likely to occur. But it can. You could have two fuse together to form one embryo, just like you have one embryo separate to form two components. So this,
So you could theoretically have two eggs, two sperm, all four of those in one person?
yes. Usually that results in death, but okay, the slow block to polyspermy.